Next. So I had two questions to put forward to the group. One was on using these, present some examples of the DBO biological data as well as the PAC-MARS data, and then to try to put forward some questions on how we may use these products for ecosystem forecasting and how we can enhance the connections between observing and modeling. Next. So on the next series of slides that you will see when this moves forward, you haven't seen it yet? Yeah, so if you don't know what the DBO is, on some of our modeling colleagues, it's a program to the, to look at the, taking from biology to, and the physical drivers. If you go back, yeah, there you go. And so we have some core measurements that are made, temperature and salinity, currents that are made on multiple cruises that go through the Pacific sector from the Northern Bering into the Chukchi and now into the Beaufort. Standard measurements decided by the participants were chlorophyll, nutrients, carbon products, plankton types, benthos, you can read this. We do annual marine mammal surveys, and then periodically fish acoustics and bottom trawling. At the same time, there are gliders, mooring arrays, sail drone for acoustics, and satellite observations. And so you can see the red boxes there. Next. And so the core mentioned were on that first slide. And if you look at the next slide that comes up, these are additional measurements that are done on the multiple cruises that provide data sets within the DBO and other process cruises. We have optical studies for satellite calibration, CDOM for dissolved organic matter and interfaces between field and NASA products, sea ice tracers, ocean acidification, and you can see measurements of fish and epifauna, genetics, moorings, and then recently measurements that we are collecting on joint cruises for harmful algal blooms. Next. And so some of the products that are available, these behind these figures that you will see next are data sets that are sitting in data archives. These are some of the satellite products that we have. If you go on the DBO website that I show you at the end, then these are NASA products. This is an example for some work from Karen Fry, basically over 4 years, looking at the timing of sea ice, which has been dramatically reduced in the last 5 years in the Northern Bering and into the southern. And if you look at this next slide, you'll be, can you move it? Thank you.
I think there's just a bit of a delay.
An example of some of the, it's just a delay. Yeah, that's fine. But are you able to hear?
Yes. So
yeah, what you're seeing here are just some of the products that are available from there's temperatures, chlorophyll on the upper right. Lower left are some of the measurements from integrated chlorophyll to some of the sediments. And on the lower right are the some of the survey measurements from NOAA on marine mammals and marine mammal types. And the base, all these maps have data sets that are sitting in the DBO data archive. Next. So if you look at the next slide, you will see is an example also of time series. So this is some of Russ Hopcroft's from UAF's zooplankton work on the left, differences between warm and cold years. And on the right are the time series that we have for some of the benthic measurements in publication that came out that we occupy these same sites over time. And some of the changes that we're seeing relate those to enhanced productivity and changes in export production. Next. Got it? So within this, within the DBO, we also have as we ramped up on the observing system, we had a large synthesis project called the Pacific Marine Arctic Regional Synthesis. And there were a multitude of PIs. You can see their names on the lower right. And we had support from Shell and ConocoPhillips for this, and the North Pacific Research Board, and through NSF. And this was one of the products that came out. And behind each one of those little figures are data sets. And these synthesis products were put forward to the local community and are also available on a website. Next. So just for example, if you go on the different themes that were under PACMARS, these data sets, that is the website at the Earth Observing Lab. It also has been transferred to the Arctic Observing site, the Arctic Data Center, excuse me, at Santa Barbara. But behind each one of those dots are various data sets, both of those data archives that would then become the basis of, this is an example of just bottom water temperatures, but we also had stable isotopes, and you can see the list of parameters on the left. Next. So on just an example of this is work that Steve Okinen, made synthesis products. These were data sets from the last 1970 to 2012. And then we were looking at CI's primary production, currents, winds, and bathymetry, and then made various data products that were brought forward and data files that are available for analysis, such as through modeling activities. Next. So when we moved on to the second theme, we had activities that addressed the phenology, or the timing of production, and how important food supply is for both the zooplankton and the benthos. And Ashton is on the line. One of the things we looked at were the hotspots and concentrations, and subsequently have been involved with one of her postdocs on a modeling activity, which Zhuan Zhang, on looking at why these hotspots retained over the times that we have, and what potential forcing functions can change that. And so the data sets, and I noticed on a previous call that Patty Matrai and previous funding by the NSF had some of these synthesis products for the on a pan-Arctic basis. So the third theme on the PACMARS was to look at some of the impacts that it has, or some of the patterns that you can see in the, I think this is the benthic one, and you would be able to, if you move from the hot spots to the next one, it just gives a summary of the 35 years of data sets that would be on the next panel if you saw it, but there it is. It was just an example of some of the data sets that are sitting. As I said, these are macrofaunal data sets that are done. We archive that is available for any type of analysis. And on the upper right is looking at carbon supply. And then next, the fourth theme when it comes up was to move the prey base to upper trophic levels. And so we're Kathy Kulis from Fish and Wildlife, Sue Moore from NOAA were involved in looking at densities of seabirds and prey, spatially in those different hotspots. Next. So when we move from this, I just wanted to close out a few examples once it comes up, to remind you to move in, addressing some of the conceptual models. For PACMARS, we put together an advective model in the upper right. The width of the bars are ones what we saw related to biomass. The areas of which we'd have pelagic benthic coupling on the upper left. And the lower left was to look at the advective input and seasonality of where some of that carbon is being brought forward. And you see it on the slide, but this is the work that's being done at Woods Hole as part of the modeling effort. Next. And then embedded in this also, we've been working with Sue Moore and Phyllis Stabenow and and others on the Arctic Marine Pulses model. And this is a conceptual model pulling in the advection and seasonality of the Pacific inflow and testing these different regions spatially as latitudinal increase with the DBO data sets, as well as some of the PACMAR data synthesis products that we have. Next. So to finalize, to close this up, what I wanted to do is to just show something we're working on with Standy Starkweather and the AON project, and that is using how do you assess the data we collect in the field, how do we move it through from left to right into an ecosystem forecasting. So this is an exercise we've only started in the last, within the last month, and that is how do you track what societal benefit areas, for example, food security or understanding the Arctic system, to services like healthy ecosystems, where we put in applications, and this is where I see the need, the obvious need for modeling and nested modeling because we have such different disciplines in this in the studies that we're doing in the Pacific Arctic, and also this has been expanded to the working towards this on the Pan-Arctic. And then on the right is the listing, for example, of some of the key parameters we collect out in the ocean as part of the observing program. And if you take this and move to the next slide, which is about the last one, I think the movement is you take it from the societal, as you're seeing, to services, the application parameters. And when this comes up, you got the parameters on the left. Now, what kind of observing products can you produce? And these are the time series data sets. We're working with Maria Kavanagh at Oregon State on seascapes. So this is working with NASA as well as with observing systems and her efforts on looking at the multiple parameters that could feed into these products. And then on the right is just a listing of some of the observing system where there are multiple data products coming out from the DBO, the Arctic Marine Biodiversity Observing Network, the EcoFoci project of NOAA. We have NASA products. If you go on the NASA website, there are other AON activities that DBO is embedded in, mooring arrays, and as I said, field observations. So if you have the last slide up there, Meredith, I think this is the one where I was putting together some of the questions that the group might consider in any type of discussions. How can we translate observations to the decision makers and various groups that are needing these data sets, as well as for a better understanding of the ecosystem? What are the key parameters for these time series studies? Can we use this as a test case for interfacing with stakeholders? For example, in 2018, the Polynia south of St. Lawrence Island did not form. We have commercial fish up in Bering Strait now. And then we also have harmful algal blooms, and how do they connect to the food web? So how do we take these observing products that could feed into stakeholder needs, identifying multi-stressors and connections, and then how do we move this into to utilize these products for spatial planning and ecosystem services. So you probably could just leave this up because the next slide is just the thank you, except you might move the next slide so people know that where they could go and get some of these data sets on the, for example, there's the NOAA webpage, but the arcticdata.io is where there's a DBO data page. If you go to the PACMARS website, if you click on the PACMARS there, it'll take you to a link to the EOL and Arctic Data Center for PACMARS. And then the NASA, as you can see, is the Neptune link there. And the Arctic Observing Ambon site also has data sets. So with that, I would just finish. I apologize for the bad connection, but I'd be happy to take any questions. And you might want to go back to the previous slide.
Thanks so much, Jackie. That was really great. We'll allow one or two questions for Jackie now, and then we'll move into Seth's presentation, and then we're hoping to have a good period available for discussion after both presentations. So do we have any questions for Jackie?
So I do have a question. Jackie, it was a very interesting presentation. When you talk about forecasting, what are the timescales that you're thinking about?
Well, this is interesting because I would think we originally thought, for example, that fisheries might be at 10 years out the change, and it happened in less than a couple of months. So to forecast, I would say probably in the 5 to 10 year, we have 30-year time series in some of those areas, not every place, but I would say that, if you're forecasting out in the 5 to 10 year period, it's probably the more realistic than to try to take it out to 50.
Mm-hmm. Okay, thank you.
Jackie, this is Renee Tedesco. Can you hear me?
I hear you.
Great. This was a very interesting presentation, so I want to make a couple of observations and ask a question. Number one question is, are there other similar DBO-like efforts taking place in other parts of the Arctic that would possibly present an integrated Pan-Arctic view?
To answer you, yes, we're working on that. I just came from the Nansen Legacy project over in the Barents Sea, and they are building an Atlantic DBO. So they're taking data sets that they've been collecting and embedding some of these transect lines. So to answer your questions, there are some areas that I think you could probably find 10 to 20 year data sets, but for spatially, we're just building this out. There's the Beaufort Sea. There's proposals to put some lines in the Baffin Bay. Just been contacted from NABOS about putting that line over the East Siberian Sea. So the answer is yes, there are, but there's different time scales of data collections for them.
That's great to hear, Jackie. I'll be very interested to hear about an integration in the years to come. So the first observation I want to make, and I did put this in the chat, is the National Weather Service Alaska region is in the process of redoing our table of organization, and we are standing up what's called the Alaska Environmental Science and Services Integration Center, or ASIC. Don Moore is the director, and you mentioned the need to get some of this information out to the decision makers, and that's exactly what the ASIC is being set up to do. And so we plan to be engaging with other federal agencies, not just within NOAA, but like the U.S. Fish and Wildlife Service, USGS, state agencies as well. And I think this might be a very good project to see how the ASIC would work in reality. So if I can facilitate your interaction with Don Moore, I'd be pleased to do that. And then the second observation I want to make is that I don't know if you've heard about this initiative that the World Meteorological Organization, WMO, has stood up. It's called the Regional Climate Center, and there's now one for Pan-Arctic. And it's not clear to me exactly how that virtual network is being used. I don't see a whole lot of need by a user base. We had the Native Indigenous communities engage with the first meeting that we had, and they deal mostly with the short term, i.e. weather, not climate. But I'm wondering, given what you mentioned about this 5 to 10 year look, I wonder if perhaps the Regional Climate Center might be a good opportunity to facilitate outreach to some of the decision makers that might need that information. So I'm certainly happy to discuss that with you further. Thanks very much.
Thank you, Renee. Those are some great comments, and I'd like to follow up with you when I get back to my office.
Happy to do so.
Okay, I think we'll move into Seth's presentation, and then we'll come back to time for discussion. So I'd like to introduce Seth Danielson. He's a research assistant professor of oceanography at the Institute of Marine Science, a branch of the School of Fisheries and Ocean Science at the University of Alaska Fairbanks. His research examines the oceanography, ecology, and ecosystems of Alaska, with a primary focus on continental shelf circulation and thermohaline structures and variations, and the atmosphere-ocean interactions that drive them. He produced the 2014 manuscript that proposed a new conceptual framework for understanding variability of the Pacific Arctic oceanic pressure head based on the location and strength of the Aleutian low atmospheric pressure system in the North Pacific and the strength of polar easterlies over Western Arctic continental shelves. Since 1994, he's participated in over 30 research cruises and served as chief scientist on over half of them. And these research cruises included both discipline-specific physical oceanography process study missions, as well as multidisciplinary oceanographic and ecosystem research cruises. And Seth is going to speak with you today about the work he's been doing on the Asgard project as part of the Arctic Integrated Ecosystem Research Program. So Seth, take it away. Seth, you might be on mute.
There we go. I'm muted. I'm unmuted. Do you see my desktop at this point?
Not yet. We're seeing your webcam.
All right. Let's try that.
There we go.
Okay. Great. Well, thanks so much for having me here today. I'm here to talk primarily about the rate experiments that we're undertaking in the Arctic Integrated Ecosystem Research Program. But I want to give just a little bit of an overview of the Arctic program to put this in context. And I just like to start by thanking the funders that are listed at the bottom, noting the broad swath of science partners that we have, and I apologize if I left anybody's logo off of here. The Arctic program, as we call it, has three primary funded teams. One of them is a social sciences team led by Henry Huntington, and this is the Chukchi coastal community's understanding of and responses to environmental change. There's a team led by Ed Farley and Carol Ladd from NOAA called the Arctic Integrated Ecosystem Survey, or Arctic IES as we call it. And then there's a UAF team that focuses on the growth, advection, respiration, and deposition, or Asgard, as we call it, rate measurements. So together, these three teams make up the Arctic program. And let's see, I'm having trouble moving my slide. There we go. Try that. Okay. And what we're doing is trying to answer the overall question of how will reductions in sea ice and the associated environmental changes influence the flow of energy through the Northern Bering and Chukchi Sea ecosystem. So this is in the context of the dramatic changes that have been observed for the last 40 years, represented by the plot on the left-hand side here that shows that for the Northern Bering Chukchi as a whole, the amount of time that it takes to go from fully ice-covered conditions to ice-free conditions, or vice versa in the fall, has decreased by an order of a month or more. And so this suggests that if you're a process that depends on the marginal ice zone, if you're a critter or a biogeochemical process that depends on this interaction and the time that it takes to do your thing, that all of a sudden you have a very different amount of time to do your work, and your influence on the greater ecosystem may be fairly profoundly impacted. So we entered this program with a few premises. First, that Arctic observations, as we well know, are strongly biased to the summer months. July, August, September are where the vast majority of our observations are from. Secondly, we lack a lot of information about the fundamental chemical and biological rates that mediate ecosystem carbon cycling and energy flows. So I'm talking about growth rates, metabolic rates, respiration rates, advective rates, turnover rates of chemistry in the water column, in the benthos, how all of these interact with the changing environment is really the key to being able to understand the environment, model it, and make projections. So our project was very much designed around the notion, and in collaboration with modelers, with Georgina, for example, to understand that we need to have the parameters that can parameterize and validate the models that we're going to be using for making projections. So it's a field-based program, but with an eye to the applications of the field results for the modeling. So our approach was to propose a study to go at a time of year where most of the observations are not taking, and given that the spring bloom may be the most biologically important event of the entire year, we decided to go to the Arctic in just in the springtime, just following ice retreat, get a handle on what's going on with the spring bloom, conduct surveys, conduct process studies and experiments to get a handle on some of these rate measurements that we think that we need for the modeling, and then to also deploy an array of moorings to be our eyes and ears around the course of the entire year. And so we have year-round measurements from a fairly well-equipped set of moorings from our project, and then in addition, NOAA has a large suite of moorings that they've been deploying. So together, the program has pretty well covered large domains or regions across the northern Bering and Chukchi Sea, so we can start piecing together a more integrated view of the system as a whole. So just to give you an idea of where we went on these two cruises in June of 2017 and June of 2018, this is on RV Sikuliak, and we're out there mapping distributions of all of the parameters and organisms that you see listed on the left-hand side, and we did this over the course of about 3 weeks and 2 consecutive years. But I'm really here to talk more about the rate measurements. So I wanted to give you an idea of what some of these are. Starting with primary productivity, and we had actually 3 or 4 ways of measuring primary productivity while we were out there. Traditional carbon-13 bottle incubations. Mike Lomas and Jeff Krause were out there in 2017 doing silica uptake experiments. Dean Stockwell also had a fast repetition rate fluorometer. One of them was strapped to the CTD so we could get profiles of the photosynthetic efficiency, a measure of how healthy the phytoplankton cells are and at what efficiency rate they're converting photons into biomass. Dean also had a benchtop FERF, and so he could do discrete samples from the underway system and from the profiles. And then, of course, the bottle incubations for primary productivity. So this, of course, is the base of the food web. And Lisa Eisner was out there with Dean. They were also doing size fractionated chlorophyll. So we have standing stock of the phytoplankton in addition to the primary productivity measurements. Growth rate experiments also extended to the zooplankton component, and this included things like the Russ Hopcroft's copepod hotel, where they incubated thousands of individual copepods and could look at the growth rate in terms of percent body size per day, and using the community cohort approach, those large 1 meter fish totes shown in the upper right have carboys with incubating seawater and zooplankton feeding for the course of, I think, about 5 days. So 11 cubic meters of flowing seawater on deck here on our large vessel like Sikuliak allows us to do some massive growth experiments and see how these animals are responding to the different temperatures and nutrient environments that they're living in. From the advective point of view, we have moorings with acoustic Doppler current profilers on them. So we're measuring the advection of the water masses, heat, salt, and fresh water that go by the mooring sites. These are chosen in Anadyr Strait and Spanberg Strait, just upstream and downstream of Bering Strait, to complement the moorings that Rebecca Woodgate has out there. A couple of the moorings had nitrate sensors on board, and one of the moorings in this array, and then one other mooring in the northeast Chukchi Sea, what we call the Chukchi Ecosystem Observatory, the CEO site, had these special water samplers pictured here in the lower right and the lower left. And these water samplers collect water into half-liter IV bags that are pre-pickled with some preservative. And so at the end of the year, we get the mooring back, we get the bags back with waters, and we can run them for nutrient analyses and plankton analyses. So we now have a year-round time series of nutrients flowing from the Gulf of Anadyr into Bering Strait and into the Arctic for the very first time, a year-round time series with a sample about every 2 weeks. Also, from looking at the advective fluxes, we have the shipboard measurements. In 2018, we towed an Acrobat undulating CTD vehicle that had a broad suite of optics and the normal temperature and salinity measurements, but also the Sikuliak has the underway acoustic Doppler current profiler and fisheries acoustics. So we also have measures of the water flows from everywhere the ship track went, and the biomass from the 5-frequency EK60 transducers that are mounted in the hull. Other rate measurements include the measurement of how fast organisms breathe. So we had respirometry measurements of zooplankton. These numbers that shown on the left-hand side are what we did in 2018. The numbers are similar for 2017. And this shows that even though these little tiny vials have only one copepod each within them, we're able to determine the oxygen consumption with a high degree of confidence compared to the baseline measurements, which are shown in green there. And so these rates will help us establish the minimal food ration required to survive. And in conjunction with the cohort experiments, we can determine the growth efficiencies of these animals. So these are all useful metrics for future modeling exercises. Other respiration measurements that were made were done by Brittany Jones and Sarah Hardy on animals collected from the multi-core deployments. And you can see that the two different species of clams, the Macoma and the Seripes, have significantly different slopes to their mass versus oxygen uptake rate. So depending on the complexity of the ecosystem that you're modeling, we now have the ability to parameterize multiple species. And eventually we're going to need to get to the point where we can where we can account for differences between different organisms. I think at this point, most modelers would probably agree that we're really data limited in terms of being able to specify with sufficient accuracy the differences between a lot of different species. So this goes for both the clams, and in the lower left, this is the whole sediment community oxygen consumption rate shown for, these are measurements from 2017, I believe. And then deposition is another important component of the carbon cycle. And here we're showing in the upper right measures from Andrew McDonald's sediment traps. So they've measuring the flux of particles. So we're talking about decaying phytoplankton cells, fecal pellets being captured by the sediment traps. And then shown in the photograph is the recovery of a drifting sediment trap that we deployed at a number of our process stations. And from the drifting sediment traps, we were able to resolve measures of the sinking speed and the carbon fluxes to the seafloor across the region, not only at the mooring sites. So beyond the rate measurements that we're doing, of course, there are the survey measurements, and of course eventually coming to a good understanding of why trawls can look so very different when you compare the two different beam trawl catches that are shown here, and the environments that are driving what are in these catches, and what these what the trawls might look like in the future in these different places are really the focus of where we're heading with these types of measurements. And so there's a lot more to the program than we could give in 10 minutes here. So I encourage you to visit the NPRB website, where we have cruise reports both from the Asgard cruise and the Arctic IES cruise. And I guess with that, I would be happy to stop and take any questions.
Thanks so much, Seth. That was a really great amount of information packed into a short period of time, so thank you so much. We'll open it up to questions. We'll begin if anybody has something specific for Seth, and then we can move into just general discussion to follow up on both the presentations.
Seth, that was, this is Georgina here. That was so exciting to see what a great data set you guys have been collecting, particularly the rate stuff for the models. I just wondered if work was been done to tie all those different rates to the environment. Like, so do we have rates that will relate to the temperature or the light, so that we could potentially extrapolate as conditions change?
Yeah, so everywhere that we're doing these process studies, we have full water column profiles with a very well tricked out CTD. So we have the light sensors. This Kuliak also has a lot of underway sensors. So we've got incoming solar radiation. We have through flow, underway nitrate measurements. So yeah, all the environmental data that goes along with the experimental work is available and there. And of course, a lot of these rates are temperature dependent. So some of the experiments were done at single specified temperatures in this Kuliak's walk-in environmental chambers, but we couldn't be changing those temperatures on a regular basis. So they were set. We have two walk-in temperature chambers, so we really had two different temperatures for a lot of the experimental work. But you know, known relations, and we can start scaling some of those with temperature as well.
That's excellent. Very excited to get my hands on it.
Yeah, thanks.
while we're waiting for others to come up with their questions, one thing I want to let everyone know is that during the Alaska Marine Science Symposium the last week of January in Anchorage, there will be a workshop for the Arctic Integrated Ecosystem Research Program on Friday morning from 9:30 to noon. And so we really encourage anyone from the broader community to come hear a bit about our program in more detail. And we intend for a lot of the time there to be open for just discussions with the PIs about potential new collaborations.
Danielle, this is Christine Mataya at NASA headquarters. I have a follow-up question for Jackie that I probably should have asked when she got done. But do you mind if I raise it now?
No, sure, go ahead.
So I just wanted to ask Jackie if it looks like the NASA DBO site covers 5 of the boxes, not 8. Is that right? Do you know if that's right? And, you know, should we talk offline about making sure we've expanded our locations if we should be doing that?
The answer is yes and yes. I think right now there were the 5 lines, but since we've expanded into the Beaufort with the changes in the Beaufort, I would be great to have an offline conversation to expand those products, because we also have our international colleagues that have access, you know, Sato and others that are doing satellite product evaluation. So it would be a real asset to be able to expand that NASA input to those 8 lines.
Okay, great. I'll touch base with folks here. I'm a contractor, and if there's any sort of financial implications, I can't make any promises or anything, but I will reach out with folks and get us connected properly to see if we can go ahead and do that.
Right, I think that's great. And also Karen Fry, yeah, you're welcome, was working with Joey Camiso at one time. So I'll also talk to Karen offline and we can connect with you. How's that?
Great, that's perfect. Thanks a lot.
Well, I have the line and maybe a connection. I would say, Seth, it's great to see those rate measurements. We also, Christina, who will be at the meeting in January, we've been doing 2 temperature rates on the DBO1 area as well as the 3, so we can, and using some of the same system. So this will be a great compare and contrast and expansion of that rate measurement on those bivalves.
Yeah, that's great. And hopefully you can both stay for the Friday workshop.
That's the plan.
I know after part 1 of this series, we heard that the modelers face some challenges in using biological data because of lack of variance estimates. And I know that often the observational scientists are having to make some tough decisions about how much sampling they can fit in with their limited ship time, and the trade-off between doing samples of replicates at given stations or being able to cover more ground. So I wondered if anyone had any thoughts on that to share.
I think that some of the newer sample techniques that we have are giving us a better way to get a handle on that. We know that the ocean has been really undersampled in terms of, say, the patchiness of phytoplankton, but having that towed acrobat vehicle that we can pull behind the vessel, we're able to resolve a lot of structure that we never would see with just the stations that we're able to stop at. Of course, that doesn't get you all of the water data and the chemical data, but it's a good step in the right direction. So once you have enough of those types of high resolution surveys, you can really start to do some good analyses of the spatial variabilities and what you're actually representing.
Yeah, this is Jim Thorson here, HEPR lead at the Alaska Fisheries Science Center. I agree that it's interesting to try to address the optimal sampling design, and there's a group of people here that are working on that for our bottom trawl data, and hopefully we can have more advice about that for fish sampling in the future. I wonder if there's a similar question for measurements of rate processes, in lab experiments. I haven't seen any, optimization questions about, given phylogenetic variation or correlations in traits, how to set up an optimal, lab experimental design to, estimate rates for the, the full set of taxa that we might want to consider. I don't know if that's the direction you were going, Danielle, by asking about variance, or you meant specifically about, field measurements of densities.
I think either is relevant, and I think it'd be useful to hear from the modelers about, what the priorities might be that they'd like observational scientists to consider as we're designing, plans to sample DBO, et cetera. What would the modeling community really love to see us focus on as a priority?
Well, yeah, as I say, for instance, in the ocean acidification program here, trying to get a better handle at, what's the full sampling frame of taxa, and, what's the physiological responses we've already measured, and how we can best optimize future lab designs to improve that knowledge, I think is something I haven't seen people work on yet.
Yeah, as a modeler, I think that's a really tough question to say which is perhaps more important. Understanding the variability in the standing stock biomasses and distributions out there, incredibly important for constraining the models. Often we're working off 1 or 2 point data points, and we get heavily criticized if the model doesn't hit those. But of course, there's a lot of variability in those data points. So it would be nice to have the uncertainty around those constrained a little bit more. And what Seth was saying. So Seth, on the kind of measurements on the undertow thing, that includes nutrients as well as temperature?
We're mounting a SUNA on that, but we haven't had that yet.
Okay, so that would be incredibly valuable.
Yeah.
Yeah, and obviously your understanding the variability in the rates is important too. But I think from my perspective, the observational data that we use to validate the model output would be sort of first order. But I as a modeler, I have no idea how, sort of the time and money that is required for that.
Yeah, I can say something too here. This is Claire Ideal. Like coming to the priorities, there's really a list of uncertainties that would be useful. And I think to starters, what the uncertainty that you're aware of to make that available. Things like detection limits, the accuracy, reproducibility, spatial variability, those kind of things, measurement, reproducibility. So it's hard to pick a priority. It's kind of, but what no matter how uncertain, it's very useful for us to know.
Yeah, we've got 2 or 3 groups making primary productivity measurements on some of these cruises, and we're going to get just as many answers to the productivity at a single station as we have groups making those measurements.
That's exciting. Very cool.
Yeah, that will be something that we'll have to spend some time wrapping our heads around. You often do not have the luxury of multiple realizations of productivity from of the phytoplankton from different perspectives like that.
Oh yeah, that's great. Yeah, do the comparisons of the methods. Yeah.
Hi, this is Lori Juranic at OSU. I just wanted to add that I've been making measurements of net community production from surface underway, mass balance of dissolved oxygen argon ratios. And I'm very much looking to make those data available to the modeling community as a constraint on integrated ecosystem metabolism. So you get a net community production in terms of oxygen, but that's easily converted into, whatever currency you're modeling in, nitrogen, carbon via stoichiometry. We only get observations in the surface, but we're getting them with averaging at sub-kilometer scale. So you can really start to see some of these features and how they're coupled to the physical variability in the system too. So if anybody wants to connect on how I can make these data available for modeling efforts, please contact me.
And I should mention that Lori was out there with us in June, so her measurements also are within the suite of what we collected in 2018 here.
Seth, is the data that's been sort of processed already available for people that want to look at it for models or not yet?
So it's still, most of the data is still in processing, and we still have one more field season. Some data sets are complete, like CTDs from 2017. I don't know. I guess we'd have to check in with the biologists as to the state of a lot of that. Danielle, do you have any thing to add for that?
I'll just add that you can contact the PIs directly for specific data sets for now. All of the data collected by the Integrated Ecosystem Research Program will be publicly available July of 2021, so that we can release a call for proposals for the synthesis phase in fall of 2021 with the intention to have that begin in 2022. So at that point, all the data will be easily publicly available. In the interim, we have an AUSE workspace that's password protected for formal collaborators, where data are shared, and some individuals may be very happy to share data in the interim.
Is there a list of who owns what data sets so we would know who to contact?
There's not currently. If you go to the Arctic Integrated Ecosystem Research Program website, you can find our integrated work plan and a sense of the data that are being collected and who's involved. And you can always contact me or Seth to be put in touch directly with folks.
Yeah.
We'll work on getting it on the website.
And the cruise reports are a good resource as well.
Oh yeah.
Absolutely.
Are they on, where are they, Seth?
They are right there at the NPRB website.
Oh, they are. Oh.
Yeah.
Oh, on the NPRB website, not the AU site.
Yep.
Yeah, the NPRB Arctic Program preliminary results page.
Cool. Great.
And so I should mention some of our projects that are interested in collaborating with us are also sharing their cruise reports there, and we're happy to add others' cruise reports. So Karen Ashton's NSF funded project, you can find cruise reports from her work there as well if she's interested in collaborating with us. So I know time may be getting short, so one thing I want to make sure to mention is I'm really excited that we've started this discussion among the modeling community and the observational scientists, and the co-leads for all of the IARPC collaboration teams will be getting together in February to make our plans for the webinars for the next 12 months and the next year. So please let us know if you have specific ideas about webinars you'd like to see us put on to continue this discussion. And Wilbert, it looks like at some point you might have had something to add to the conversation. Do you want to jump in?
Yeah, I guess I have two questions. One is one of the colleagues of mine, Scott Elliott, and Georgina is also involved with his work and aware of his work. He's very much interested in marine organics and on the surface layer and how it affects the air-sea transfer. Did you guys have any measurements on that?
No, not specifically on that. Although, and I don't know whether or not this is relevant, but in the photograph you see there, Eric Collins is swabbing the mooring that just came back, and he was doing microbial analyses on sort of all the samples that he could get his hands on, water samples, surface water samples from depth. And after going out with us on the Ambon cruises and these cruises, he identified 5,000 or 6,000 new species that hadn't been previously identified in these waters. And so he's got, these data have, the analyses have been run through the genetics put into the database, so he can start pulling out the genes of the various critters that are responsible for, say, different types of nutrient turnovers. And so there may be some linkages between what he's doing and that type of approach that you mentioned.
So does it have to do with the macromolecules, the big lipids and?
Well, it's in terms of the microbes that could be breaking those down, yeah, and the reorganization of them, yeah, I would think.
Okay, and I guess another question is to follow up on the variability question. And I'm not a marine biogeochemist at all, so this is a question out of ignorance, I suppose. But to what extent is the variability that you see in the ecosystem structure, for instance, at any given location, is that controlled by the environmental factors, basically? And to what extent is there also a lot of internal variability in the ecosystem structure and itself, basically?
I think there's a lot from both directions. I think a lot of the structuring in this area is driven by the physics of high nutrient waters being put through very turbulent, constricted straits that provide nutrients up to the sunlight where productivity can take place, and then you have a huge export of carbon to the seafloor. So we have all these hot spots around that Jackie likes to study. And so there's a very bottom-up driver of the system, but then you also have ecosystem dynamics such as the whales and the walrus that have the ability to eat themselves out of a foraging spot for a while and have to go find someplace new to forage in. So there are top-down effects as well.
Georgina, how's that in the kind of models that you're running? Do you have any insights on that? To what extent the variability is really reflective of the environmental variability?
Well, I mean, so spatially and seasonally, the models generally capture those sort of gross trends in variability. As a rule of thumb, models never capture the full extent of the environmental variability. You know, right. Sort of our error bars around sort of the nutrient concentration at a location over a month is never anywhere close to what they observe. That's sort of what I find. We're sort of close to getting mean trends, but we don't get the highs and the lows.
Right. Yeah, that makes sense. So there's a lot of variability just in the internal structure, basically, of the system. Yeah.
That's right, and that's what, particularly the coarse resolution models, is not great at capturing.
Right. So that's definitely a problem then, I suppose, for
1 degree model.
Yeah. All right. So thank you.
We do have about 5 more minutes for this discussion period, if anyone else has further questions.
So I guess one question that I have, how much are you guys involved in observing marine biogeochemistry in the sea ice? Do you have any kind of dedicated research programs to make observations in sea ice?
Well, I would say, this is Jackie, there have been people like Rolf Grodinger, who's now in Norway, doing sea ice. They're particularly in the early spring period. But within our observing program, we're not there. The ice has been gone, went out at the end of April this year and hasn't even come back yet. So the sea ice part of this, looking at the biogeochemistry within the DBO program is not there, but within specific process program has been done periodically, particularly around Barrow and previously during the Bering Sea Ecosystem, the BEST program. So there are data sets, but they're limited.
Okay, great, thank you.
Yeah, and for the IARP program, it's not an ice field directed program. We considered that, but it wasn't in the cards to be able to afford all of that. The only way we can really get at it is from the perspective of the moorings. And for instance, the mooring up by Hannah Shoal, we have some really beautiful year-long records of PCO2 and pH, and the relation between the storms and the PCO2 and then the ice and its evolution over the course of the winter is really interesting. I know that there'll be a poster at AGU in a couple of weeks showing some of that off.
Okay, cool.
Seth, I don't know if the sediment traps and water samplers on the moorings that can capture flux on a monthly basis might give you some information that you could draw from. As the ice trees.
They definitely do, and the mooring that we've had up at Hannah Shoal has particularly high resolution, say 5-day sampling from April through June, specifically to try to capture the export of ice algae when the ice is melting. And we see those signals showing up very clearly in late April and early May, even before the ice, 1.5 months before the ice has even gone away. It's, some very interesting signals.
Do you have fluorometers on this one?
And we do have fluorometers on some of
And
those.
I would also add that the issue of variability as well as sea ice is that there are two sediment traps and moorings in the Southeast Chukchi Sea. And there's a recent paper out by a new postdoc, Waga, who looks at sea ice, the similar type of thing that Seth is talking about being seen in the southern Bering, southern Chukchi Sea, early before the ice is even gone. But what I'm saying is there's a couple of moorings that have sediment traps, so we could look at some variance aspects there, as well as we can look at some variance in the benthos too, because we do multiple reps at different regions. So I would say for me, a variance study would be focused on one of the DBO sites and not across the whole 600 kilometers.